All right. We are live. The link is posted on Discord. And we are ready to go. So today we're going to be doing something a little bit unusual. We're going to be reading a biology paper. Not really reading, but more so trying to understand what it is. So there's a science paper that came out recently. Let me make sure that we have volume. Okay, we do have volume. There's a paper that came out recently that kind of made a bunch of waves, and it's called Loss of Epigenetic Information Drives Aging. And the kind of TLDR is that epigenetic changes are now being blamed for aging, and we have a variety of mechanisms to change the epigenome. So maybe that's kind of a way to modulate aging in mammals, and then thus in some long distant future potentially have immortality. So I'm not a biology expert, so obviously when I try to read this paper, I am constantly running into words and terms, especially all this chemistry stuff that I do not know what these are, right? So rather than sit here and then struggle through a paper, I've thought that there's an interesting thing we can do here, and what we can do is we can actually try to read this paper in tandem with some big language models. So here I have a Hugging Face open. So Hugging Face is kind of a machine learning, maybe community is the right word if you want to say it, but it's basically just a space where people can post different things. In particular, the feature that I'm using here is called Spaces, which are ML apps that are made by a community. So you can see there's different things here. You can have text to audio. There's a bunch of image ones. There's, I don't know, any kind of weird ML model you could think of. But the ones that I am particularly interested in, or the ones that we're going to be using today, are called BioGPT Large and Big Science Bloom. So Big Science Bloom is a large language model that has been trained and created by the community at Bloom, which is an open source community. And I'm currently running this on a T4 medium GPU, so it's costing us about 90 cents an hour, but that's more than fine for the price that we're going to pay. And this is what you can do with Big Science Bloom, is you can basically put questions in. So what is animal hair made out of? And then you put it in this kind of QA format. And one thing you can also do is say, let's think step by step. And then it'll basically autocomplete for you. First, what is hair? Hair is a protein, is keratin. Keratin is a protein, so you can basically then copy that, put that in there, submit that, and then there you go. It'll just basically, it'll keep completing what you put in there. So you can change different parts of what it is, and over time you can get a better definition. So let's see what it says. It says, first, what is hair? Hair is a protein called keratin. Keratin is a fibrous protein that is found in the skin, nails, and hair of animals. Okay. So we could also ask the same thing of a different large language model. So this other large language model here is BioGPT Large. So we can check out its model card to figure out what it is. And this is a pre-trained language model that has been fine-tuned on biomedical stuff. Domain-specific generative transformer language model pre-trained on the large-scale biomedical literature. So Big Science Bloom is a little bit more generic. I think it probably won't have good answers for some of the more intense questions that we ask it over the course of this stream. But I think BioGPT Large is going to be a little bit better because it's fine-tuned on biomedical journals, so it should give us slightly better answers. So you can do the same kind of QA formulation here for it, but the output's a little bit less clean. It's a little bit more raw. But let's see what it says. What is animal hair made out of? The hair shaft is made of keratin, a fibrous protein. The hair shaft is made of keratin and the associated proteins. The hair shaft is made of keratin and the keratin associated proteins. Okay, so it kind of got stuck in this A, B, C, D, E kind of thing. Let's maybe we can get better at prompting. Animal hair is made out of, let's see what it submits there. Okay, animal hair is made out of keratin, a fibrous protein that is similar to keratin found in the skin of other animals. The hair is a natural fiber that is used in a variety of applications. Hair fiber is made of complex structure, lipids, blah, blah, blah. Okay. So finally, just because we don't want to be just asking these large language models and not necessarily double checking, we're also going to have a window here where we have Wikipedia, and we'll be able to type in, for example, keratin, and then see what Wikipedia has to say. So with these three separate tools combined, right, we have Big Science Bloom, we have BioGPT Large, and we have Wikipedia. What we're going to try to do is we're going to try to understand the abstract for this paper, right? And it's just a couple paragraphs, but it's incredibly dense material, right? So that's the mission for today. Let's go ahead and get started. Epigenetics changes are hallmark of aging, but whether they are the cause or consequence of aging process in mammals was unknown. Now, a study published by Cell showed that inducing changes to the epigenome causes physiological and molecular signs of aging. So let's start off with what is the epigenome? What is the epigenome? A. Let's see. The epigenome is the sum of all the chemical modifications to the DNA and histone proteins that, let's put that in there, submit again. It'll autocomplete past that. That are not encoded in the DNA sequence. These modifications are important for regulating gene expression. Okay. Why don't we copy paste the epigenome is and put it into BioGPT Large, see what that has to say about it. So BioGPT has to say the epigenome is a dynamic structure that is regulated by the interplay between DNA methylation, histone modifications, and non-coding RNAs. So both Big Science and GPT have brought up histones. So let's see what those are. The epigenome is essential for the maintenance of cellular identity and is involved in the regulation of gene expression, cell differentiation, and cell fate determination. The epigenome is also involved in the regulation of cell proliferation and apoptosis. So here, more terms that we don't necessarily know here. The dysregulation is associated with the development of cancer. So histone modifications in epigenetics. Histone modifications are, let's see. I think we can, let's see, we might be able to not use this prompt here, prompting style. Okay, so Big Science Bloom. In epigenetics, histone modifications are the epigenetic regulation of gene expression is a dynamic process that is regulated by a complex network of, I think this one really wants the Q&A. What are histone modifications? Let's see what this guy said. In epigenetics, histone modifications are important for the regulation of gene expression, are regulated by histone acetyltransferases and histone acetylases. Damn. These are divided, HDACs, okay, are divided into 4 classes. class 1 HDACs are ubiquity expressed, while class 2 are tissue specific. So why don't we actually search for histone on Wikipedia and see what we get there. So a histone acetyltransferase, which is actually, that is what BioGBT was talking about here. Histone acetyltransferases, HATs, are enzymes that acetylate. In organic chemistry, acetylation is organic esterification reaction with acetic acid. It introduces an acetyl group into a chemical compound. Such compounds are termed acetate esters or simply acetates. Okay. So these enzymes, called histones, acetylate conserved amino acids on histone proteins by transferring an acetyl group. DNA is wrapped around histones. Okay, so I think this is the key part, is that the histones are the molecules on which the DNA gets wrapped around and kind of compressed. Okay, genes can be turned on and off. By transferring an acetyl group to the histones, genes can be turned on and off. In general, histone acetylation increases gene expression. So how does this happen? Let's ask Big Science Bloom. How does histone acetylation increase gene expression? Okay, we'll copy paste that in there. Copy paste this in here. Okay, the acetylation of histones is associated with the activation of gene expression. The acetylation of histones is associated with the activation of gene expression. The acetylation of okay, so you see this? Big Science Bloom here got stuck in a little loop, and it's just kind of repeating the same sentence to us over and over and over again. So what if we just get rid of this first sentence, see if it can find a second sentence? Now it gets stuck on this first sentence. Okay, so let's copy paste this answer that it gave us and put it into Big Science, or BioGPT. I think BioGPT will probably be a little bit more powerful here. Okay, here you go. The acetylation of histones is associated with the activation of gene expression. The acetylation of histones is regulated by histone acetyltransferase and histone deacetyltransferase. Okay, and then it went into the kind of into a similar kind of thread that we saw before, where it goes and it lists the different HDACs. In general, histone acetylation is linked to transcriptional activity and is associated with euchromatin. Euchromatin, which is less densely compact, allows transcription factors to bind more easily to regulatory sites on DNA, causing transcriptional activation. Okay, so I think I have a little bit better of an idea about what histones are, what acetylation is, and how histones can be acetylated in order to associate and cause certain types of gene expression, because they're basically sitting there and the DNA is wrapping around them, right? If you change something, transfer an acetyl group to some of these histones, you can turn genes on and off. So that seems to be the TLDR of how histone acetyltransferase interacts with DNA in order to alter the epigenome. Okay, so let's go here to the next paragraph. To test the long-standing question of whether epigenetic changes can cause aging in mammals, the author used a mouse model termed irreducible changes to the epigenome, ICE, that expresses the slime mold endonuclease I-PPAL in a tamoxifen-inducible manner. Okay, that is fucking gnarly, so we're going to type that in there. The slime mold endonuclease I-PPAL in a tamoxifen-inducible manner. Let's see what Big BioGPT has to say about that. Okay. The anti-estrogen tamoxifen, TAM, is a potent inhibitor of the growth of the slime mold Physarum polycephalum. The effect of TAM on the activity of endonuclease I-PPAL, a DNA cleaving enzyme of the Physarum polycephalum, was investigated. TAM inhibited the activity of endonuclease I-PPAL in dose. Okay, so endonuclease I-PPAL, a DNA cleaving enzyme. Let's see what that is. Let's see if Big Science Bloom knows anything about this. Describe endonuclease I-PPAL, a DNA cleaving enzyme. Let's think step by step. First, what is endonuclease I-PPAL? Let's copy paste that, see if that helps it. Endonuclease I-PPAL is a DNA cleaving enzyme. This is a type of enzyme that breaks the phosphodiester bond. Okay, that seems like it's getting on the right little train of thought there. Let's go ahead, let's let it continue doing that. The phosphodiester bond between two nucleotides in a DNA molecule. It is a type of endonuclease. Let's see if we copy paste this into here, if it gives us anything. Oh. Oh, we lost it. Copy. It is a type of enzyme that breaks the phosphodiester bond between two nucleotides in a DNA molecule. It's a type of endonuclease. It's a type of endonuclease. It's a type of... Okay, this got stuck in this pattern. So that's actually weird. We copy pasted the output of Big Science Bloom. And again, Big Science Bloom is not fine-tuned on like biomedical stuff. So it's just kind of more of a generic large language model. We copy pasted the output of that into BioGPT, which is fine-tuned on biomedical stuff. So this thing has a little bit more of an idea about biomedical stuff. And it got stuck in a loop here, which is kind of interesting to think about. Like why did this get stuck in a loop? Okay. Let's see if we just ask it to talk about endonuclease I-PPAL. See what it comes up with. Endonuclease I-PPAL is a DNA cleaving enzyme that recognizes and cleaves the phosphodiester bond between the two bases of sequence 5, pi-pu-pi-3. Okay. The enzyme has been used to cleave DNA at specific sites in the genome of bacterium E. coli. The cleavage reaction is highly specific and does not occur at any other site in the genome. Okay. The enzyme has been used to cleave DNA at specific sites in the genome of yeast. Okay, so it basically seems like they're using this I-PPAL to catalyze DNA double-stranded breaks. So basically, it's a way of cutting DNA molecules. So that's what this I-PPAL is doing. Okay. So previous research, now we're back into the abstract here. Previous research has shown that the cellular DNA break repair process involves chromatin modifying enzymes and results in epigenetic changes. Okay, let's type that in. Cellular DNA break repair process involves chromatin modifying enzymes and results in epigenetic changes. Involves, and then is enzymes written wrong here? Enzymes. Enzymes like this. Okay. Let's see what BioGBT has to say. Cellular DNA break repair process involves chromatin modifying enzymes and results in epigenetic changes. The epigenetic changes are mediated by histone modifications, DNA methylation, and non-coding RNAs. The epigenetic changes are also involved in the regulation of gene expression and cellular differentiation. Okay, and then it gets stuck in a little pattern here. Okay. Let's clear it out. So, why don't we see what chromatin modifying. Let's type that in there. Chromatin. There we go. Chromatin is a complex of DNA and protein found in eukaryotic cells. The primary function is to package long DNA molecules into more compact, dense structures. Okay, so it's similar to the histones, is basically what we're learning here, is that you have your DNA, and then you have your histones. So your DNA gets wrapped around the histones. So these are the histones that we learned about earlier. So the first step is basically wrapping around these histones. This is called the nucleosome. Then you have beads on a string. Okay, so then basically your long strand of DNA is going to be wrapped around some amount of histones. Okay, there seems to be a difference here between core histones and then histone H1. So maybe it seems like there's different resolutions of histones. These seem like larger histones, and then you have smaller histones. So histone H1 is larger histones. Add further scaffold proteins. Okay, more scaffolding proteins, more scaffolding proteins, more scaffolding proteins, and then you finally get to the actual chromosome shape. Okay, so I'm guessing that chromatin is one of these scaffolding proteins that are kind of on the end here, or that like are towards the end of the structure. The primary function is to package long DNA molecules. This prevents the strands from becoming tangled and also plays an important role in reinforcing the DNA during cell division, preventing DNA damage, and regulating gene expression and DNA replication. So it seems like kind of what we're learning here is that a lot of how the epigenome, which is which genes are expressed, right? You have your DNA, which is your genome, which is what code actually exists in your DNA. But then only a subset of those are going to be expressed, right? And basically you can think of it like some genes are turned on and some are turned off. And epigenome is basically the config file for your genes. It's which of the genes are actually on and which of them are off. And it seems like the epigenome is largely comes down to these histones and chromatin, basically these different types of proteins and enzymes upon which everything is wrapped and everything is packaged, right? So basically the packaging of the DNA is what results in specific parts of the DNA being activated and not. The primary protein components of chromatin are histones. Okay, so chromatin is a histone, it seems. Or chromatin is a superset of histones. Right? It's an octamer few repeating units which could be derived. So it's basically composed, chromatin is basically composed of a bunch of histones. You have histone 2A, H2B, H3, H4, so there's basically seems like a couple different types of histones here. Bind to DNA and function as anchors around which the strands are wound. Okay, so let's go back here to our abstract and see if we can understand this a little bit better. Previous research has shown that cellular DNA break repair process involves chromatin modifying enzymes, so enzymes that modify the chromatin, right, that are messing with these histones, that are messing with basically the wrapping of the DNA. In isolated ICE cells, which are basically mouse cells, the authors confirmed that immediately after IP-Pol induction, and IP-Pol is basically going in there and breaking up these histones, DNA breaks are faithfully repaired. Cell cycle, apoptosis, and senescence are unchanged. Okay, so let's apoptosis and senescence. Let's see if Big Science can answer that. In biology and genetics, what do apoptosis and senescence mean? A. Let's think step by step. And the let's think step by step is basically just to force it to like kind of have a linear train of thought. This is like a trick, a prompting trick that people have come up with, where if you force it to, if you give it this in your context, it'll force the big language model to be a little bit more precise. Okay. Apoptosis is the pre-process, a process of programmed cell death. It is a normal process that occurs in all cells. It is a process that is essential for the development of the embryo and the maintenance. Okay, what if we just remove that last part, because it's probably just going to go into embryo stuff, and see if it goes into senescence. No, it's very convinced. So this is interesting, is that this language model, like with the same context, it'll give you the same answer. So usually there's something called temperature, where you can modify how much curiosity versus kind of greedy optimization this will do. So it's interesting that it does that. Let's go over here. Let's ask BioGPT large. In biology and genetics, apoptosis and senescence, or in cellular biology, apoptosis and senescence refer to, and let's see what it thinks. Okay, in cellular biology, apoptosis and senescence refer to two distinct forms of cell death that are characterized by distinct morphological and biochemical features. Okay, so Big Science Bloom basically told us that apoptosis is programmed cell death, but BioGPT large told us that actually apoptosis and senescence are both different types of cell death, but they're different. Apoptosis is a highly regulated process that is characterized by shrinkage, chromatin condensation. Okay, so we just learned that chromatin is basically the scaffold upon which all the DNA is wrapped. So chromatin condensation means the chromatin, I guess, condenses out, it precipitates out of it. Nuclear fragmentation and the formation of apoptotic bodies. In contrast, senescence is a state of irreversible cell cycle arrest that is accompanied by a series of morphological and biochemical changes. So it seems like apoptosis is when the cell actually is told to die, right? So apoptosis is the cell is getting a signal to destroy itself, and it goes through a kind of a regulated process that very slow, that basically says, okay, we're going to shrink this cell, we're going to get rid of all the chromatins, we're going to take away all the scaffolding, we're going to break up this DNA, and we're going to basically destroy the cell. Versus senescence, and these are called sometimes senescent cells, is when the cell has kind of gotten into what's called like a zombie state, right? Where it's basically, it's no longer doing what it's supposed to be doing, but it's largely just kind of sitting there and doing nothing, right? So let's see if we can confirm what we just learned on Wikipedia. Cellular apoptosis and senescence. So let's click on both of these articles, see what we get. Apoptosis comes from ancient Greek, which means falling off, is a form of programmed cell death that occurs in multicellular organisms. Biochemical events lead to characterizing cell changes and death. Okay. These changes include blebbing, cell shrinkage, nuclear fragmentation, chromatin condensation. It's interesting that if you actually look here, these exact steps are what BioGPT gave us, right? Cell shrinkage, chromatin condensation, and nuclear fragmentation. Cell shrinkage, nuclear fragmentation, chromatin condensation. They're in a slightly different order, but it's basically the same thing. Here we have a prostate cancer cell exploding into a cascade of apoptotic bodies. Okay, so you have this cell here. It seems like it's undergoing apoptosis, and this purple part of it gets very large, and then it basically just breaks up into little droplets of nothing, which I think are what apoptotic bodies mean. Okay. So what is cellular senescence? Cellular senescence is a phenomenon characterized by the cessation of cell division. In their experiments during the early 1960s, Leonard and Paul found that normal human fetal fibroblasts in culture reach a maximum of approximately 50 cell population doublings before becoming senescent. Okay, so it basically seems like, here, let's look at this. The Hayflick limit deliberates that the average cell will divide around 50 times before reaching a stage known as senescence. As the cell divides, the telomeres at the end of the linear chromosome get shorter. The telomeres will eventually no longer be present on the chromosome, the end stage of the concept that links the deterioration of telomeres to aging. Okay, so I think we've all heard about telomeres before, but telomeres are basically like the very end of your long DNA strands, and they're basically just like buffer space, right? So that every time a cell duplicates and basically copies its DNA, it's going to cut a little bit off the end. So you basically have a bunch of padding at the end so that you can keep cutting and duplicating. But over time, right, your telomeres are going to shrink, and then eventually you're going to start cutting into DNA that's actually doing something. And it seems like senescence is what happens when the telomeres finally run out. The physiological importance for cell senescence has been attributed to prevention of carcinogenesis and more recently aging development and tissue repair. Senescent cells continue to contribute to the aging phenotype, including frailty, sarcopenia, muscle loss, frailty, which is like kind of bone density loss, and aging-associated diseases. Senescent astrocytes and microglia contribute to neurodegeneration. So basically, senescent cells are what's causing mammals to age, right? Like the frailty, loss of muscle, neurodegeneration, all these things come from the fact that the cells that make up these tissues are becoming senescent. There's a larger and larger percentage of senescent cells. So it seems like what we want is we want cells to go through apoptosis as opposed to senescence, right? We don't want them to just become zombie cells. We want them to be told to die so that you can have nice fresh cells. So if we go back to our paper here, DNA breaks are faithfully repaired. Cell cycle, apoptosis, and senescence are unchanged, and the epigenetic age increases. Okay, so they basically take these mouse cells, they put them in this solution, or in this iPeopal solution. And this actually, I don't think it's an L. I think it's IPPOI. All right, but they put them in this crap, and this crap basically breaks the DNA. And then once you have this DNA that is broken, right, and it's broken because the chromatin was modified, right? The scaffolding upon which the DNA sits was broken, so the DNA starts having all these little breaks. And then once you have all these little breaks, the DNA breaks start getting repaired, right? So how can we get this repair to happen without the breaking, is I guess where this is going. The researchers then induced the expression of the enzyme in adult mice for 3 weeks and comprehensively inventoried behavioral, psychological, and molecular features of aging 10 months after tamoxifen dosing. These included gray hair, decreased bone density, right? So this is the frailty that we were reading about here. Frailty, a decline in memory, that's neurodegeneration, and then reduced movement, which is, I guess, sarcopenia. The researchers also mapped the epigenetic landscape and measured epigenetic aging. They observed that the epigenetic landscape in ICE samples was eroded, leading to a loss of cellular identity and an increase in epigenetic age. Okay. So basically they took normal mice, they broke their epigenomes by messing with the chromatins, and then after breaking the chromatins, even though the DNA was repaired, the mice themselves started to become old and started to exhibit the characteristic kind of problems of aging, where basically they have reduced bone density, neurodegeneration, and sarcopenia. So I think the important thing here is that apoptosis and senescence are unchanged, right? That sentence there is really the key, because it used to be, I think what they're trying to argue here is that a lot of people thought that cellular senescence and kind of the relationship of senescence and apoptosis were maybe driving biological age. But what they did here is that apoptosis and senescence were unchanged, and then the mice still got very old. So they only got very old because of the epigenetic age. So this kind of, by only changing the epigenome and seeing the mice get old, they're basically confirming that the epigenome is what is causing the age here. Jose Diaz Caldero. Hello. Okay. So let's ask here. Is epigenetic changes responsible for neurodegeneration and sarcopenia? A. Let's see. Epigenetic changes are a major contributor to the aging process. So it does have an idea, right? It seems like Big Science Bloom, like, it does think that epigenome is important. Defined as heritable changes in gene expression that do not involve changes to the DNA sequence. Epigenetic changes, and then it's probably going to repeat that sentence. Okay, so it kind of gets, Big Science does understand that epigenetic changes have a contribution to the aging process, and then it knows roughly what epigenetic changes are, but it's probably not going to have the same context. Let's see what BioGPT large has to say. Epigenetic changes are responsible for neurodegeneration and sarcopenia. Let's see what it auto-completes here. The aim of this study was to investigate the effect of exercise on the expression of genes involved in the regulation of muscle mass function and the brain of aged rats. Okay, so it's like starting to... This BioGPT large was fine-tuned on medical journals, so it's not like you're asking it a question, right? It's basically just saying, given this sentence, what is the most likely second sentence, right? So because it's been trained on a bunch of scientific papers, it tries to hallucinate a scientific paper with everything that you give it, right? So here, it's basically taking this term here and then just starting to like hallucinate the paper that it would have read it from. So I don't know if that's necessarily going to help us, but epigenetic changes. Are epigenetic changes? Let's think step by step and see if we can get it to not just hallucinate a study. Okay, no, now it's like kind of hallucinating a weird like question paragraph. Epigenetic changes will result in neurodegradation by modifying the epigenome. See? We can potentially prevent or delay the onset of those age-related diseases. Okay, so there you go. BioGPT Large also believes that epigenome is responsible for age-related diseases, and that by modifying the epigenome, we can eventually get rid of these age-related diseases, which is largely what this whole epigenetic aging paper is about. The team also demonstrated that cellular reprogramming, which is known to rejuvenate the epigenetic landscape, can counteract ICE effects. Let's see what that cellular reprogramming can rejuvenate the epigenetic landscape. Let's see. And restore their ability to differentiate. In this review, we discussed the role of epigenetic mechanisms in the regulation of pluripotency and differentiation, and how these mechanisms can be manipulated to generate induced pluripotent stem cells. Okay, let's see what Big Science has to say about cellular reprogramming. In epigenetics, cellular reprogramming refers to, or what does cellular reprogramming mean? Cellular reprogramming is the process of changing the identity of a cell. All right, keep going. It is a process that is used in the laboratory to create induced pluripotent stem cells. Okay, so let's search for induced pluripotent stem cells on Wikipedia. Induced pluripotent stem cells, also known as IPS or iPSCs, are a type of pluripotent stem cell that can be generated directly from a somatic cell. Somatic cell is a biological cell following, so just kind of your average everyday normal cell. The iPSC technology was pioneered by Yamanaka and Takahashi and Kyoto, who together showed that the induction of 4 specific genes, right? These 4 specific genes. I wonder actually, Yamanaka factors. Do you think that Big Science knows what the Yamanaka factors are? Let's see. I'd be very curious to see if it actually knows what it is. The Yamanaka factors are 4 specific genes. What are the 4 Yamanaka factors? The Yamanaka factors are OCT4, SOX2, KLF4, and CMYC. MYC, OCT4, SOX2, and KLF4. So look at that. That is pretty crazy. Somewhere within the weights of this giant big science bloom, it knew this perfectly. It encoded the exact Yamanaka factors. That's pretty crazy to think about. All right, let's see if I feel like if Big Science Bloom got this question, it's 100%, or the BioGPT large is 100% going to get it. We'll prompt it kind of like this. See if it completes that. Okay, the Yamanaka factors are for specific genes. OCT4. It's interesting that both Big Science and BioGPT said OCT4, but when you go to Wikipedia, there's OCT3 and 4. So there's a couple different types, it seems, but OCT4 seems like the one. SOX2, SOX2, SOX2. I'm looking at the order here too, right? So it's interesting that BioGPT and Big Science both have the same order here, right? They have OCT4, SOX2, KLF4, OCT4, SOX2, KLF4, and CMYC, CMYC. What's also interesting is that these are, the capitalization here is different from the capitalization here. But both of these got it correct. The Yamanaka factors are expressed in the nucleus of a cell and are responsible for the reprogramming of the cell into a pluripotent state. The Yamanaka factors are also known as the Yamanaka factors because they were discovered in 1998. Okay, so basically you have these 4 different factors, which are genes that can cause a cell to become a stem cell, and the closer you are to a stem cell, the further away you are from this epigenetic aging, right? Pluripotent stem cells hold promise in the field of regenerative medicine because they can propagate indefinitely as well as give rise to every other type of cell. They represent a single source of cell that could use to be replaced those lost to damage or disease. So that's the key here, right? And it's not. So basically, this is how these people think that they're going to achieve immortality, right? Is that we are going to go ahead and we're going to say the reason you're old, the reason you have low bone density, reduced muscle mass, and memory decline is because the cells in each of those tissues have bad epigenomes, right? The epigenomes for those cells has kind of gotten out of control. Your little chromatins and histones, they've all gotten all tangled up and they're broken, and it's leading to problems with those cells. So what we're going to do is we're going to basically use these 4 Yamanaka factors. We're going to, I don't know, inject you with them or somehow like put them in a virus and then put that virus inside you. I don't know exactly how you're going to get these inside the cells, but they're going to put them inside your cells. And what they're going to do is they're going to basically take your old cells with broken epigenomes and kind of pull them towards a stem cell. So they're going to take that brain cell that's sitting in your brain and has a very messed up epigenome, and they're going to try to make it more of a stem cell. And by making it more of a stem cell, what's going to happen is the epigenome will kind of fix itself, right? It'll become more like a stem cell, and then it'll become a brain cell again. So it's kind of like a reboot, like you're rejuvenating that individual cell and rebooting it into a stem cell so that it can then differentiate back into a brain cell with a nice, clean, brand new and fresh epigenome. So that is kind of the process. I don't think they have any way of actually, yeah, here you go. They use a viral delivery method to express the Yamanaka factors in the mouse cells. So kind of predicted that, which is kind of cool, but I mean, it makes sense. So basically they take a virus And inside this virus, they put the 4 Yamanaka factors, and then those 4, basically whenever the virus goes, and normally viruses, what they're trying to do is they're trying to go into a cell and then just inject their DNA. So viruses act as a very useful mechanism for injecting DNA and molecules into cells. So this treatment reduced epigenetic age. Okay, so they're almost there. So they took mice, they messed up their epigenome with this IPPOI. Because of their messed up epigenome, the mice aged faster and looked way older. And then they put in a virus that had these 4 Yamanaka factors in it, and the epigenome of those mice got younger. Okay, the last part that we're missing is that the mice whose epigenome was messed with and now the epigenome is rejuvenated should hopefully have gotten some of these things fixed, some of the memory, the movement, and the bone density fixed, but there's no sentence for that, so it's just a little bit dubious. Like, why didn't they say that, right? That seems like that would have been the nail in the coffin. These results show that the changes in the epigenetic landscape are sufficient to drive aging and support the idea that epigenetic reprogramming poses a potential therapeutic avenue for the treatment or prevention of age-related diseases. However, further research will be required to understand the mechanism behind epigenetic aging and rejuvenation and determine whether the epigenetic landscape can be safely restored in humans. This is awesome, dudes. This is like, we're close, because it seems like we're not far from a future where you can get an injection, right? An injection that's filled with viruses, and these viruses have all these Yamanaka factors, and then those Yamanaka factors go and they go everywhere in your body and they start winding the clock back, right? They start restoring the epigenome of all the different tissues in your body, and then thus effectively making you younger, right? So this is immortality here. I think the problem with these type of things is usually that I'm sure there's like kind of a cancer trade-off. So a lot of times these kind of rejuvenation mechanisms, what happens is that they also cause cancer. So it'll be interesting to see if they can do it without, I think that's the word, safely there. Pretty cool. So we kind of understood this, guys. This is a good idea. This is a good point here. We took this abstract, which was absolutely chock full of words and terms that we didn't understand. We fed it into BioGPT Large, which is a large language model that's been fine-tuned on publication data. And we got some pretty good explanations out of BioGPT Large. We fed it into Big Science Bloom, which is a more generic large language model. But both of these language models actually knew what the Yamanaka factors were. They knew what senescence and apoptosis and histones and chromatins. So these large language models were very helpful in helping us understand this abstract. And then we also double-checked on Wikipedia, right? Wikipedia is edited by humans. So this Wikipedia is going to be, I don't want to say 100% correct, but it's going to be much more correct than these big language models. Because these, you don't know exactly. It's kind of like a black box. It might not be correct. But after this experience, I'm kind of convinced that actually I don't need to go on Wikipedia. Like the fact that both Big Science and BioGPT Large, both of them got the 4 Yamanaka factors perfectly, that is pretty crazy. Okay, so one more thing I wanted to do before ending this stream here is this. So this is a figure from the paper. So you have to pay money to get the paper, and I wasn't going to pay money to these crusty scientific institutions to get the paper. I'd rather pay a Hugging Face for an hour of these models. So let's try to understand this figure with everything we've learned. Okay, so we have young and we have old. So we have an old mouse here. Let me actually move this here so you guys can see a little bit better, because I think my face is blocking it. Yeah. We have a young mouse and we have an old mouse, and then we have like a little clock here. So they're saying the epigenetic clock in the young mouse is younger than the epigenetic clock in the old mouse, right? So these two different mice have two different kinds of epigenomes, the old epigenome and the young epigenome. They're kind of using this epigenetic landscape, kind of ball rolling on a hill analogy here, where I guess in the old mouse or in your old epigenome, the ball can kind of move around more freely, versus in the young epigenome, it's much more stuck in a specific thing. So I think maybe what they mean here is that in an old cell, in a cell where the epigenetic landscape has kind of gone to shit, the cell isn't activating the right parts of the right genes, right? So basically, like, the cell is producing all kinds of weird proteins that don't necessarily help for the type of cell that it is, versus a young cell, a cell where the epigenome is correct, is much more focused, and it can only really do one thing. It's only expressing the genes that are very specific to that cell type, right? So you have youthful epigenome and epigenetic entropy. Let's ask what epigenetic entropy is. All right, let's see here. Epigenetic entropy is a measure of the degree of randomness in the distribution of the number of different types of mutations in a tumor. It is a measure of the degree of heterogeneity of the tumor, and it is a measure of the ability of the tumor to evolve. Okay, so this is like, it seems like it kind of thought we were in like a cancer context. Let's see what Big Science has to say. What is epigenetic entropy? Entropy of the epigenome, which is the set of let's copy that, autocomplete, which is the set of epigenetic marks that are present in a cell. The epigenome is a key regulator of gene expression. Okay, so epigenetic entropy, I guess here you have high entropy, that's what they're showing you, and then here you have low entropy, right? Entropy means randomness, chaos. So chromatin contacts. Here you have kind of pictures that show that the chromatins are only contacting with maybe specific little regions that are local to them. But then here in the old cell, you have chromatin that is contacting parts of the epigenome that are, I guess, further away from each other, right? So what are chromatin contacts? Chromatin contacts are physical interactions between two genomic loci, so two places in your genome. They interact. They can be detected by chromosome conformation capture. Let's see what BioGPT has to do with this. Chromatin contacts are mediated by the DNA binding domain of the TF and the DNA sequence of the binding site. The DNA sequence of the binding site is referred to as the recognition motif. The recognition motif is represented as a position weight matrix, PWM, okay? Matrix of the probability of each possible nucleotide of the motif. Okay, so that's probably what they're showing you here, is that chromatin contacts for the old cell are kind of broken, and you see, whereas here previously the chromatin contacts, all three of the little arcs were right on top of each other. Now some of the arcs are kind of going to the wrong places. So you ideally want this, which is more differentiated, more narrow, less entropy in your epigenetic landscape, And once you get old, your chromatin contacts go all over the place. You have more entropy in your epigenetic landscape, and that's bad. All right, cell identity and X differentiation. So X differentiation. X in the context of epigenetics, X differentiation refers to, let's see what BioGPT large says. In the context of epigenetic, X differentiation refers to the process by which a cell acquires a new phenotype without any change in the DNA sequence. And again, all cells in your body have the same DNA sequence. This process is important in the development of cancer, where it is thought to be a key step in the progression of tumors. Okay, so there's a lot of cancer papers. This thing has been training a lot of cancer papers. It really wants to get to cancer papers. Let's see what Big Science has to say. In the context, okay, wait up. The context, what does X differentiation refer to? See, it might be better if we do that. In the context of epigenetics, X differentiation is a term coined by blah, blah, blah. Okay, and then it's probably just going to explain what this dude is, so I don't want that. So H3K27AC. Because we have no idea what that means. H3K27AC is a histone modification that is associated with an active gene expression. Okay. And H3K27Me3 are, let's see. In the context of epigenetics, H3K27AC and H3K27Me3 are considered to be marks of an active and inactive chromatin, respectively. Okay, so this red part here, H3K27AC, is active chromatin, and this lower row here, H3K27Me3, is inactive chromatin. So this is showing you where the chromatin is inactive, and this is showing you where the chromatin is active. We found that the levels were significantly increased in the promoter regions of the gene encoding the transcription factors in the livers of mice fed a normal diet. Okay. All right, so I think I finally understand what's going on here. So in the young cell, right, so in this part here, this column here, which is all the young cell, the chromatin, right, which is the scaffolding upon which the DNA is put on, is very specific about what it is in contact with, right? The chromatin contacts are very specific and very narrow. So you see here how H3K27AC, which is active chromatin, is very high, right? Like it has a high spike here, which means that it's very strong affinity for these regions and very strong negative affinity, right? So inactive chromatin, it's very strongly repelled against these regions. So this is showing you that the chromatin is very opinionated in a young cell, and that it very much wants to bind with these areas and very much does not want to bind with these areas. So now, if we switch into an old cell, so this column here represents an old cell or an old mouse, right? A cell in an old mouse. Now, the chromatin contacts, they're a little bit more all over the place, right? They're no longer very opinionated and narrow. Now they're kind of just contacting all kinds of random things. We see here that the active chromatin sites, right, are lower, right? The height of these red spikes here and the height of the red spikes here is different. These are lower, which basically means that the chromatin isn't very sure anymore, right? It's not super certain about where exactly it wants to contact. It's the kind of positive contact signal and then the negative contact signal are reduced. So it's kind of the this analogy of the ball on the hill, right? It's like the spikes are lower, right? It's like the activation and the high points and the low points are a little bit more similar, which means that it's easier for this ball to roll around on this epigenetic landscape. So that's basically what they're showing you here, is that these specific marks here, which are telling you where the chromatin is active and inactive, they're much less, I guess, opinionated or much less clear in the old cell compared to the young cell. So maybe one thing that I don't understand here is OSK. So let's go ahead and ask BioGPT that. In the process of cellular reprogramming, OSK is... Let's see. In the process of cellular reprogramming, OSK is able to induce the expression of endogenous Yamanaka factors. The expression of these 4 transcription factors is essential for the maintenance of pluripotency. Okay, so it seems like OSK is what leads to Yamanaka factors. Let's see if we can get it to actually define. OSK stands for OSK stands for OSK. Oh, okay. OSK is literally the Yamanaka factors. It's the first 4 letters. OCT4, S for SOX2, and then K for KLF4. Okay, so now we know what this means. So OSK here basically just means Yamanaka factors. And by using that viral delivery mechanism to change the epigenome of the mouse cells, you can basically revert all of the cells from their old epigenetic state, where it's kind of all loosey-goosey and everything's kind of falling apart, and reprogram them into a younger epigenetic state where the chromatin contacts are much more opinionated, the cell is much more opinionated, it's more differentiated, it has a more stable cell identity and a more functional tissue as the result of that. So this is good. I think we achieved our mission. We are a little bit smarter and we understand epigenetic aging a little bit more. Yeah, thanks to BioGPT Large and Big Science Bloom for helping us learn about all this stuff. So I'm going to go ahead and turn off these machines. So this is these large language models that we've been using. They're running in a cloud provided by Hugging Face, but I don't want to keep paying 90 cents for all the hours, so I'm going to change them to CPU basics, confirm new hardware. These are free, so I don't have to pay. So I'm going to go settings, CPU basic, confirm new hardware, and factory reboot the space. Alrighty. That's it, dudes. Thanks for listening. Hope you learned a little bit. Hope you can maybe use LLMs to understand stuff that you guys are trying to figure out. And if not, thanks for listening. Like and subscribe, and tune in next week for another kind of series of 3 live streams. Thank you and goodbye.